So we're going to go into a little more depth on the molecular basis for inheritance. So today's topic begins our conversation more about chromosomes. We've talked about chromosomes a little bit before this point, but the chromosomes we're going to look at, we're going to deal with them as molecules, we're going to deal with them as physical bodies, and kind of look at how their behavior in the process of cell division and other processes, how that kind of sets down the rules for what we've already observed in Mendelian and other forms of inheritance. So first off, a little background on what a chromosome is. So the word chromosome refers to a colorful body, chromo for color. These chromosomes in this image are artificially colored in a technology that really didn't exist for a long time. So it's a relatively recent technology where they can so-called paint the chromosomes with a certain color of fluorescent molecule that highlights certain gene regions. So these are specific to gene regions found on chromosomes, and the unique pattern for each chromosome allows you to identify it. So it's kind of a quick, if you got the money to do this kind of technique, a rather quick way to identify your chromosome 1 and chromosome 2, etc. So this is from a human. And this highlights the 23 different kinds of chromosomes that humans have. And you can see the sex chromosomes in humans are the 23rd pair. This individual has an X and a Y, making them a male individual. And also highlights the fact that the other chromosomes exist as pairs. So these are our autosomal chromosomes, and they are paired in an individual. So this adult human has 2 of chromosome 4, 2 of chromosome 5, and so on. So these are homologous pairs making the individual diploid. So diploids are really characterized by having this homologous pair of chromosomes. So where do we find chromosomes? I've mentioned that we can find them in the nucleus of a eukaryotic cell, and that's mostly where we're going to consider finding them. But I think since we're doing a genetics course, I think it's important to acknowledge there are other places, even in eukaryotic cells, where we can find chromosomes. So, for example, the organelles in eukaryotic cells have a little bit of their own DNA. And those DNA is arranged in chromosomes, not the same form as what we just saw, but it is described as a chromosome. So if we look at the animal cell, kind of standard animal cell, standard plant cell, inside of this nucleus here is most of the DNA, and certainly most of what we worry about in this genetics class. But also in the animal cell, the mitochondrion has some DNA in its genome and its chloroplast. The chloroplast, in its chromosome, the mitochondrial chromosome. The chloroplast has its DNA in a chromosome inside of this organelle. And so those are little bits of genetic information that do belong to these organisms, even though it's a rather small proportion of the total. DNA also found in every other kind of living cell, or every living organism has DNA as its genetic material. So if we look outside of eukaryotes and into a prokaryote, this bacteria, then the DNA is found inside of the bacterium. It's not inside of a nucleus, but it is present inside of the cell, sort of loosely organized in this illustration. So what I mentioned about bacteria, kind of interesting because that has a tie-in to what we see in eukaryotic organelles. So the mitochondrion and the chloroplasts that are found in eukaryotes, these are actually organelles that have an origin in ancient ancestral times, long billion years ago. They were their own free-living bacteria. And you can see this as a similarity in the DNA that makes them up. Number one, it's organized into this circular chromosome. And number two, the genes, if you were to compare their DNA sequence, the DNA sequence of the human mitochondrion is much more similar to the genes present in a bacterial genome than the mitochondrial DNA is to the host eukaryotic cells. So some good evidence for the relationship of these to ancestral bacteria, chloroplast and mitochondrion. I mentioned previously, there's some genes located on the mitochondrion that they don't typically come up in genetics class, but if you were to have a mutated version of one of these, this could cause a rather severe phenotype, so-called mitochondrial disease. So they're a small component. They're relatively rare that we talk about these from a genetic standpoint, but they are very important genes found on the mitochondria. Okay, so before we get into eukaryotic cell division and chromosomes, just another illustration of bacteria, and using this to kind of start us in on the topic of cell division and dividing chromosomes. So the process of one bacterial cell dividing into two involves splitting up the DNA, the genome of that starting cell, into the two resulting cells. So if you're going to have one cell make two cells, then the starting cell that goes into cell division has to have twice as much genetic information so that a full set can go into one cell and a full set into the other cell as a result of cell division. So an important prerequisite for cell division is DNA replication, and that's shown happening here, where the squiggles of DNA in the first cell are shown as being twice as much in the second cell. One way to keep track of this, I know they're not easy to tell apart, one way to keep track of them is that there's a single origin of replication marking that bacterial chromosome, and so there's one on this copy and one on this copy, and those are actually used to pull these chromosomes apart. So bacterial cell here shown as having one chromosome. That's pretty common in bacteria. Just a single circular chromosome with all the information this cell needs to get its stuff done. Now, if we look at the eukaryotes, a lot more complexity in the organism and also more complexity in how its DNA is organized. So lots more DNA, as we will see, in eukaryotes relative to bacteria. And so it's organized a little more carefully. So if you look at the naked double strand of DNA, this molecule comes in at a rather small size, and it's organized by wrapping it around specific proteins into a structure called a nucleosome, and then those are wrapped together into a tightly bound structure, and then these are wrapped together, and then those are wrapped together. So it's a several orders of packaging to get that single molecule of DNA wrapped up into what we see as a chromosome. Now this chromosome on the right, this is kind of your typical chromosome shape, this kind of X shape. We'll see what exactly that represents in a little bit. This is large enough you could see it with a certain light microscope. So high-powered light microscope would show you chromosomes, and scientists use that actually to count chromosomes. The other levels, looking at the actual DNA plus nucleosome structure, this is something you would have to see using an electron microscope. But these different levels of organization, we understand them pretty well. A lot of packaging. So why package up your DNA? Well, DNA is packaged up the same way you might pack your clothes for a trip or for moving, is that you need to be able to move a lot of loose stuff in a compact fashion. So the cell has to move this chromosome, even though it's made up of lots of loose squiggly DNA, it's got to make it over to the side of the cell without getting caught on anything or confused for anything else. So these are packaged up so that they can be moved. Now, while they're packaged up, they're not very useful as far as using those genes for giving the instructions for making proteins and so on. So the packaging of DNA is very brief from the standpoint of the whole cell cycle or the lifespan of a cell. The packaging happens in eukaryotes at the beginning of mitosis, so right before cell division, and the DNA is already loosely packed again by the end of mitosis. And mitosis is a small part of the overall cell cycle. So this form of, a little more familiar form of chromosomes as they appear here, is relatively rare, but it does help us to see them as distinct units separate from each other. So this DNA shown in the center could have come from an individual that also had cells that look like this before mitosis. And so this is just blue staining the DNA, green staining microtubules. And inside of the nucleus, you can see the nuclear shape is pretty well outlined by this blue mass. And there's not a lot of structure to that. So it's mostly loose strands of DNA present in the nucleus. And this is most of the cell's life. So having the the DNA loose like this. Similarly, if you had your clothing unpacked, this allows you to choose an outfit for the day, to see all the clothing options you have available. Same thing with the DNA. Having the DNA loosely packaged allows enzymes to get in there and copy the genetic code to be able to make proteins and use other functions of DNA. So difference here, but we tend to think of our DNA in these chromosome units. They're easier to draw, they're easier to think about, I think, and we can kind of point to locations a little bit better on those. So you'll see me drawing chromosomes like this. Just keep in mind that there are lots of times in the cell's life when the DNA is actually loose, like it's shown in the left image there. Another image of cells dividing. This is an onion root tip, and that's a region that people often use to look for cells undergoing mitosis because the cells there are rapidly dividing. So a little better chance of finding the chromosomes packaged up as they are in those two cells that are highlighted. And still plenty of cells that are just going through growth parts of their cell cycle, and their DNA is loosely packed with a circle outlining the boundaries of the nucleus. Okay, so we mentioned in the bacteria, the DNA has to get copied before it can get divided into two cells. So the process of a cell growing, copying its DNA, and dividing, that's called the cell cycle. And cells go through this pretty much as rapidly as they can when they are actively dividing. So for example, if you took a yeast cell and put it into fresh medium, gave it a fresh food supply and no competition, it would start growing rapidly and working its way through this cell cycle as quickly as it can. And so I want to highlight a couple features on here. These are the abbreviations for the different phases of the cell cycle. G for growth or gap phases. S for DNA synthesis. So this is the process in this phase of the cell cycle where the single set of chromosomes becomes kind of a double set. And I'll explain that in a little bit here. And then during the process of mitosis, that double set gets split apart again. So I have some questions on here. What are we looking at when we look at these cells? So we have a number of chromosomes, and it's not a trick question. The number of chromosomes are the number of distinct bodies you can count here. So 1, 2, 3, 4, 5, 6, 7, 8. 8 chromosomes there. And if we go up to the cell after DNA synthesis, we still count 1, 2, 3, 4, 5, 6, 7, 8. So these chromosomes are bigger, they're twice as big, but each one is still a chromosome. So we're going to use a terminology later that I'll show you that calls these replicated chromosomes, and these ones down here would be unreplicated chromosomes. Apart from that, they are still the same number of chromosomes. So there are 8 distinct chromosomes, and we know this individual is a diploid because each chromosome has a homologous buddy. So these exist as homologous pairs, and a homologous pair is a pair of chromosomes that are the same size, the same length, the same centromere position, and then on there would be the same genes in the same order. And so as we've seen in inheritance genetics, we've seen that individuals that are diploid get an allele from dad and an allele from mom for the same gene. So this individual diploid, 8 chromosomes total, but you would look at this also as 4 pairs of chromosomes. And then after replication, 8 chromosomes existing in 4 pairs. So this terminology here of replicated and unreplicated chromosomes, I think this is something that doesn't often get covered, and I think it really should be covered a lot more. Something that I think helps to really understand the terminology of chromosome and also kind of what you might be looking at any one point. So overall, I'm hoping that you can kind of make this make sense for yourself as you go through the process of learning about chromosomes and their behavior in cell division. So shown on here would be a single chromosome comparing its unreplicated version on the left with the replicated version on the right. And again, kind of confusing, or maybe it doesn't sit well with people, this idea that the thing on the left is a chromosome and the thing on the right is a chromosome. They're both still one chromosome. If you want, you can use this terminology of chromatid. Chromatid refers to kind of like one of these linear pieces of chromosome. On the right, this is a chromosome made up of two chromatids, and on the left would be a chromosome that is only one chromatid. It can get a little confusing, and I'll hope to use some terminology that just doesn't, I don't want to confuse you if I can help it. Other features that are highlighted on here are features common to chromosomes. The centromere, as we'll see, has a function in keeping these two replicated halves together, and also in pulling them apart when that happens during mitosis or meiosis. Centromere is not always at the exact center. Sometimes it's offset. Sometimes it's practically at the end of the chromosome. And the ends of the chromosomes are called telomeres. These have special structure that keep them from degrading over time. So here's a little video of centromeres in action showing their function. And it really is, at one point, they need to hold these sister chromatids together. That's kind of the first half of the video on the left. And at another point, they have to let go and let these sister chromatids go to their own directions. So what's shown on the left here, these are microtubule fibers that are pulling the respective chromatids apart and then toward different ends of the cell. This is a part of the mitosis process. And on the right is a live image of a plant cell showing the chromosomes, a whole bunch of chromosomes all moving away. Those circles that are highlighted, those are the centromeres. And you can kind of see how they are the anchor point where these chromosomes are being pulled apart. So the chromosome arms are kind of flailing out as like loose spaghetti behind, but they're getting pulled from their centers or centromeres. You don't have to know any of this detail, but I thought you might be interested to know a little bit more, or maybe just to see about chromosome centromere structure. So the centromere has a certain DNA sequence that the microtubules and associated proteins can recognize. So it has a little bit of a signal on there that says, I'm a centromere, you need to use this area to pull me apart or hold me together. And there's a whole bunch of proteins, that's what's abbreviated here. All these little units here are enzymes that are responsible for holding the centromere together, attaching it to microtubules and so on. Okay, so we saw unreplicated and replicated chromosomes. This is a situation where we have a homologous pair going from unreplicated to replicated. So both of these show homologous chromosomes. The two colors, you can think of those as mom's version and dad's version. So a diploid individual gets one of the homologous chromosomes from dad and one from mom, and passes only one of those on to a gamete. So unreplicated to replicated, we still have blue and orange. And again, here we have sister chromatids within one chromosome, and then homologous chromosomes are chromosomes that look like each other but are not physically connected to each other. So to go from one to the other, if you wanted to go from unreplicated to replicated, that's a result of DNA replication, otherwise known as S phase, or DNA synthesis is what the S stands for. And then to go back from replicated to unreplicated, that's what happens during mitosis when these chromosomes are pulled apart at the center, and each cell that results gets one copy. So it's sort of a switching between these two forms of chromosomes as the cell cycle goes on. All right, so if we're looking at a gene, we've looked at genes before, and we're pretty familiar by this point with the idea that individuals get two alleles for a gene. And this is a little more of a molecular look at that. If you have a gene with two alleles, then you have the two homologous chromosomes, and each chromosome has a copy of that gene. So you can have a different version. You could be heterozygous and have dad's version be different from mom's version. But after replication, whatever you have on dad's version gets replicated and copied exactly onto the other chromatid of dad's chromosome. And same thing for mom's chromosome. So if you look at this as alleles, then you can look at this as an individual that's heterozygous. The blue chromosome gets a little b, the orange chromosome gets a big B. After replication, think about this for a second. After replication, the little b is across from little b. So this blue chromosome has only the little b version. This orange chromosome has only the big B version. And we get those from copying the starting cell. I'm sorry, the starting chromosome. Okay, so looking at the process of mitosis, I've been using the term mitosis and talking about cell division. If you are really into your vocabulary, you might have felt a little uneasy about that. Basically, in this class in genetics, I'm going to use mitosis and cytokinesis as one event. So mitosis technically is nuclear division, and cytokinesis or cell division is the division of the cell. Quite often it happens that nuclear division is followed by cell division, and we're just going to treat it that way. So I'm not going to throw the terms, really going to kind of avoid the term cytokinesis, but just keep in mind that that's sort of buried into what we're talking about here. So the end result of mitosis is an exact copy. Mitosis happens in cells that are copying themselves exactly. So if you're a single-celled organism, like a yeast cell, that would be one cell making two exact versions of itself. Mitosis also happens during the growth of multicellular organisms. So if a skin cell produces another skin cell, then those cells are genetically the same at the end of this process. So your cells, you got a whole bunch of cells that have a version of your nuclear genome, and they all have the same version of your nuclear genome because they were produced by these exact copying events of mitosis. So we start in with two short chromosomes, two long chromosomes, a red and a blue short, a blue and a red long chromosome. And an important step in mitosis, it's important to remember that you start mitosis with copied, replicated chromosomes. So we go with two long and two short, and we still have two long and two short, except now going from G1 here to the mitosis, they are now replicated chromosomes. So replicated long red, replicated long blue, replicated short red, replicated short blue. And the specific phases of mitosis, I'm not concerned that you memorize those. You've probably done that in previous courses. I don't need to make you do that again. So I generally focus on metaphase as the phase when we can really see the genetic results of one of these division events. So in metaphase, you can see how the long red and the long blue, they're lined up along this central, what they call the equator of the cell, the midpoint of the cell. And they're not attached together, and they're not even associated close with each other, but they're each going to get pulled apart. So the two replicated halves of this long red chromosome get pulled apart to opposite ends of the cell. So long red goes here, long red goes there. And that happens for all four of these. And then the chromosomes that go on the left here go into their own cell. The chromosomes that go to the right get into their own cell. And we end up at the end with two cells that have chromosomes that look just like what we started with. So two short, two long, short blue, short red, long blue, long red, short blue, short red, long blue. All right, same stuff, now two cells instead of one. So this process of mitosis is important for making sure that the resulting cells get exactly what the starting cell started with. All right, so you could take this and draw these chromosomes out, solve your own mitosis problem.